Hi, my name is Andrew Hsieh. I'm a physician scientist at the Fred Hutchinson Cancer Research Center in Seattle, Washington, USA. I'd like to thank the organizers of this meeting for giving me the opportunity to share some of the work that's being done in my research laboratory. This work was done by a team of scientists in my lab, led primarily by Yi-Ting Lin, who's a postdoctoral fellow. So in my lab, we've been very interested in studying how the 5' UTR is actually deregulated in cancer. And that's because we know the 5' UTR can coordinate gene regulation at multiple levels, in particular at the transcript levels within the DNA, and at the transcript level, at the RNA level, particularly influencing transcript stability and translation. So our central hypothesis is that mutations within the 5' UTR should be able to tune gene expression in the context of cancer and actually afford cancer additional ways to deregulate gene expression. We know this in part through work that's been done by other laboratories, because when we look at the 5' UTR and it's full of regulatory sequences, we know that some of these regulatory sequences have been shown to be associated with various cancers that afflict mankind. So in our laboratory, we wanted to start from a kind of 50,000-foot point of view, looking at all somatic mutations within a particular human cancer. So we decided to look at human prostate cancer, and that's because it's the most common cancer. It's one of the most common cancers in men within the United States. It's the second leading cause of death. So in particular, what we did was we started out looking at 229 patient specimens, both localized specimens as well as metastatic specimens. And what we did was we did 5' UTR sequence-based analysis to see how often somatic mutations occur within these cancer types. Overall, we found 2,200 somatic mutations within the 5' UTR, which affected 1,878 genes. Now, looking at the characteristics of these 5' UTR mutations across localized disease and metastatic disease, we saw that the number of times mutations occur actually increase as a patient develops more aggressive disease or metastatic disease. And this is very consistent with what we see in the coding sequence. What's particularly interesting too is that while 835 of these genes that have mutations overlap between 5' UTR and coding sequence, about 1,043 or 55% of the 5' UTR mutations affect genes that aren't otherwise affected in the coding sequence, suggesting to us that 5' UTR mutations may be another mechanism by which cancers can deregulate the genome to drive aberrant gene expression. Another clue that this might be the case was just looking at the distance between recurrent 5' UTR mutations. And we saw that about 40% of 5' UTR mutations, when they were recurrent, occur within 50 base pairs of each other. So given this finding, we want to know whether or not somatic mutations within the 5' UTR preferentially affect cis-regulatory elements, known cis-regulatory elements at the DNA and at the RNA level. So in order to determine whether or not this was happening more than what would be expected by chance, we modeled 2,200 somatic mutations within the 5' UTRome and permutated this 10,000 times in an analysis. And what we found was that at the DNA and the RNA level, we saw that mutations that happen at these levels were happening way more than would be expected by chance if we just randomly put in 2,200 mutations within the 5' UTR. And this is taking into consideration the trinucleotide context of the mutation. Now, we want to know whether or not this was a class effect or was happening to specific motifs. So we looked at RNA binding motifs or RNA motifs that have been described. And what we found that it wasn't a overall effect on every single RNA motif, and there was certain motifs that are being affected. Here, we found those top-like or PRTE sequences had more mutations than we expected by chance, but this was not the case for URs, G quadruplexes, and 5' tops. So given these findings, now these are just correlative findings, we want to know whether or not the effects that these 5' UTRs could have on gene expression, both at the transcript and translation level. And in order to address this question, we created a new technology, which we called the Pooled Full-Length UTR Multiplex Assay on Gene Expression, otherwise known as plumage. And what this is a highly parallelized approach to measure the impact of 5' UTR point mutations on transcript levels and translation rates simultaneously. To do this, we went ahead and developed a method to incorporate pretty much any length 5' UTR, and that's one of the important features of this, any length 5' UTR into a reporter construct as a luciferase reporter construct. Now, in order to be able to sequence this, in order to measure changes in transcript and translation levels, we put downstream of the stop codon a 30 base pair randomer barcode as shown here. Now, in order to link the barcode to the various 5' UTRs and to pull out the correct linkages between the two, we did PacBio long-read sequencing of our plasmid library. And through this, we were able to constrain our library down to about 216,000 unique plasmids. Once we had our library identified and we had basically the address book for our entire library, went ahead and transduced these into various cell lines and then measured our barcodes at the DNA, RNA, and polysome bound levels. And what we found was that out of the 545 recurrent mutations that we looked at, approximately 35% of them had impact on transcript levels or translation rates, as shown here. And what's particularly interesting is that when we looked at these genes, we found that these affect various cancer pathways, such as the MAP kinase signaling pathway, and various cellular processes that are usurped in the process of cancer, such as apoptosis. Now, given these particular findings, we wanted to validate whether or not what we're seeing with plumage was actually occurring using orthogonal models, orthogonal systems. So at the transcript levels, we decided to focus on two genes. There's two oncogenic proteins, FGF7 as well as FOS, as shown here. These were predicted through plumage to actually upregulate transcript levels. So we went ahead and did qPCRs on these plasmids after transduction in the cells. And indeed, we saw an increase at the transcript level. What was more interesting is that when we looked at the FGF7 mutation within a patient who had that particular 5' UTR mutation by RNA-seq, we found that indeed this patient expressed high levels of FGF7 that was statistically significant. And when we looked very closely at what the mutation was actually doing at a nucleotide level, we found that this mutation was actually creating an E-box. And E-boxes are very interesting because they are the putative binding site for the MYC-MAX heterodimer, which is an oncogenic protein capable of transforming cells and very, very deregulated in cancer. So in order to determine whether or not this neopromoter actually is bound by the MYC-MAX complex, we did an EMSA, where we basically determined whether or not the mutant, but not the wild type sample, can now bind the MYC-MAX oncogenic complex. So what we can see here is that with either MYC or MAX, neither one of them bind the wild type or the mutant DNA in this assay. However, only in the context of the mutant do we see binding with the MYC-MAX complex, and this can be competed off by using an unlabeled competitor, demonstrating that 5' UTR mutations at the DNA level can create neopromoter elements that can drive gene expression of oncogenic genes. What about at the translation level? Well, there we actually saw upregulation of oncogenic proteins like AKT3 and downregulation of tumor suppressors like NUMER1. So we went ahead and did luciferase assays on these constructs individually as an orthogonal test to what we were seeing in plumage. Indeed, we saw that with NUMER1, there was a decrease in luciferase activity, and with AKT3, we saw an increase. And what was really interesting was that this was associated with ablation of SRSF binding sites, as shown here, of SRSF9 for NUMER1 and SRSF2 for AKT3. One other very interesting observation that we saw within our data set was that singular mutations can have varying effects on gene expression. For instance, the tRNA synthetase CARs, a particular 5' UTR mutation, led to a significant decrease at the transcript level, but a significant increase at the translation level. And we were able to validate this using a qPCR and reporter assay as shown here. What this demonstrates is that singular mutations can actually influence more than one level of gene expression. We need to keep that in mind as we actually study patient-associated mutations. Given these findings, which were all done in reporter systems and mass Pella reporter systems, we wanted to know whether or not in changes to endogenous sequence context of the 5' UTR, could this have an impact on oncogenic gene expression? To do this, we went ahead and used David Liu's base editing system, where he takes a highly evolved APOBEC and links it with CRISPR-Cas9 to actually lead to very precise point mutations within the genome. So we went ahead and decided to choose to study this for a particular hit of an oncogenic cell cycle regulator called CKS2. And one of the reasons why was because this mutation was about 65 bases upstream of the ATG. And this was interesting to us because the mutation that we had was from a C to a T, as shown here. And if this mutation were actually functional, would create an N-terminal extension of this coding sequence. So we went ahead and mutated CKS2 at this site within our cell line, 293s. And we were able to do this for one allele, but not the other allele. And we developed 3 different mutant lines. And in these mutant lines, what we saw when we ran Western blot and blotted for CKS2 was indeed in each one of the mutant lines, we saw the creation of this N-terminal extension at high levels without actually compromising the 11 kD normal CKS2. And this is the quantification shown here. Looking at the mRNA levels, we saw no difference. So what this shows is that even in the endogenous context, when we go ahead and mutate one allele of one gene at one particular point mutation, this can have a significant impact on gene expression of oncogenic genes like CKS2. Lastly, we were very interested in understanding what the clinical context of these mutations are in patients. So indeed, we looked at all the mutations that we had for localized and prostate cancer patients. We were very interested in seeing were there things that were specific for localized and specific for metastatic disease. Using gene set enrichment analysis, we indeed saw things that were similar, but we also saw things that were different. For example, the enrichment of MAP kinase signaling pathway seemed to be very specific to metastatic patient specimens. And this is very interesting because the MAP kinase pathway has been shown causally to, in mouse models, causally to induce metastasis. So we were interested in seeing if a patient had MAP kinase mutations, did this associate with worse outcomes for those individuals? And what we found was that patients with MAP kinase mutations were twice as likely than those who did not within the 5' UTR to present with metastasis at diagnosis, demonstrating that 5' UTR mutations actually correlate with worse patient outcomes when it's associated with particular oncogenic signaling pathways like the MAP kinase signaling pathway. So in summary, full-length 5' UTR multi-level functional genomics is achievable through plumage. Through plumage, we saw that 35% of recurrent 5' UTR mutations are functional in the prostate cancer genome. And this could be done at the level of the DNA creating neopromoters or at the level of RNA ablating RNA binding sites. There could be bi-directional effects, as we've shown with CARs, and it could lead to N-terminal extensions, which can change translational dynamics. And importantly, 5' UTR mutations may associate with worse patient outcomes. Thank you very much. I'd just like to acknowledge my lab, who put in a lion's share effort to get this work out and for doing this work, as well as our collaborators and our funding sources. Thank you very much.